It's really great to have Dr. Benjamin Voight as the visiting professor to Rutgers Institute for Translational Medicine and Science. Dr. Voight is associate professor in the Department of Systems, Pharmacology, and Translational Therapeutics, and the Department of Genetics at University of Pennsylvania School of Medicine. He received his PhD degree in human genetics under the tutelage of Dr. Jonathan Pritchard and Dr. Nancy Cox at the University of Chicago in 2006. From 2006 to 2011, he did his postdoctoral training at the Broad Institute of Harvard and MIT with Dr. Mark Daly and Dr. David Oster. In 2011, he began his academic career at UPenn, where he quickly moved up to the rank of associate professor with tenure in just 6 years. Currently, he's member of the American Diabetes Association, American Heart Association, and American Society of Human Genetics. He served as editorial board on JAMA Network Open and Circulation, as well as external referee for many journals, including Nature, Nature Genetics, Cell, Lancet, JAMA, and Science Translational Medicine. For the last 16 years, he has developed and utilized statistical genetics, computational biology, and population genetics approaches to understand the biological underpinning of evolution history of human trait and complex disease. He has published over 200 peer-reviewed manuscripts in many high-impact journals such as New England Journal of Medicine, Science, Cell, and PNAS. And in just 16 years, he has penned 14 Nature and 44 Nature genetics papers with an H index of 80. It's truly great to have Ben as a visiting professor to Rutgers. Ben, you can take over.
Great. Thanks, Steve.
Oh, by the way, this lecture is being recorded, so if you can all mute yourself and unmute yourself when you have a question. Thank you.
Okay, I think I got it now. I just course under pressure, choke, press the wrong button. Great, okay, I think I'm good here. Folks can see my slides? Perfect. Thanks for the intro. Thanks for having me virtually. Man, it's rotten. I can't be there in person. Maybe around the next time then we can do this in person when this whole Omicron pandemic nonsense ends. But great to be here and always excited to talk about Mendelian randomization. It's a fun little tool and technique, and I think has a lot of really nice potential opportunities in this kind of data-rich era that we live in. So, I feel like it's important to kind of like stage really the question and really what we're trying to get at. And really it's about causality. And I think it's easy and many of us appreciate that causal inference is, if not one of the most important, is certainly one of the most challenging problems in biology and medicine. But it's a really important one because if our objective is to make biological and clinical insights that have therapeutic potential, we really have to understand causal biology fundamentally. And what I hope to do today for those that are uninitiated is to describe this technique as generating one line of evidence for causality using human genetics data. True causal inference, those causal relationships that are really robust, require multiple lines of evidence from human clinical trials, from model systems, from cell models, from fundamental understanding about biochemistry and pathways. And we need lots of lines of evidence to support causality. What I hope to describe today is that human genetics can provide one additional piece of evidence to build the case for causality and understand the causal inference puzzle. So fundamentally, what are we really trying to get at? So let's imagine this very simple idea where we have a trait of interest. Maybe that's body mass index, maybe it's LDL cholesterol, maybe it's a circulating inflammatory marker. And we're really keen on some outcome of interest. Maybe it's a disease outcome, it's diabetes, heart disease, maybe it's a intermediate trait that we think is relevant, maybe it's a complication. But we really would like to understand if this arrow pointing from some change in that exposure modulates that outcome directly. And in practice, maybe we have some lines of evidence that support whether or not that arrow is true, but really we're trying to make inference about whether or not that arrow is actually exists or not. So the key idea behind Mendelian randomization is basically like the transitive property. So if we really believe this hypothesis that the trait actually directly modulates the outcome, then if a genetic variant happens to, if we know about a genetic variant that actually perturbed that trait, then in principle, if that's true, then by the transitive property, the genetic variant should also perturb the outcome as well, with some assumptions that we'll discuss, so stay tuned for that. But this sort of mediation pathway is really just the fundamental idea behind Mendelian randomization. This approach and this idea is pretty popular. So I took this opportunity, given this talk, to see how prolific this area has sort of grown. So I just PubMeded articles with either Mendelian randomization of the English and British spelling of Mendelian randomization in PubMed. And, you know, it's something on there of like 22 or 2,300 articles, and that's probably an underestimate because not all MR papers report doing MR experiments in the title. There's probably like work done in fundamental papers that are doing other things too. And it's actually grown pretty expansively. And if I looked really carefully, it probably, you know, if you squinted hard, you probably could see two inflection points. One is really, you know, around the order of 2011, 2012, when researchers first started getting access to large-scale biobanks like the UK Biobank, where it's just like a rich genetic catalog, hundreds of thousands of people profiled with array data across a range of electronic health records, phenotypes. So just a really rich genetic resource. So, you know, like many things, like the availability of data means the availability of experiments for people to do. But then I think the other inflection point probably picks up around 2018, where, and I'll discuss this later, where the tools to perform these kinds of experiments got embedded in a website, which basically aggregated a bunch of data as well. So it just made very accessible the approach to a lot of people. And then at that point, things have just sort of taken off, and seems to be no stop to that. I think there's already like, it's like, when are we in 18 days into 2022, and there's already like 80 articles in PubMed. So I think this has grown, and I think it's really grown just because data is prevalent and tools are prevalent. So with that kind of framing in place, today I thought I'd sort of like do the following. I'll kind of walk you through how I understand intuitively the framework, give you a little bit of background on the fundamental model, the regression model that's utilized, describe the fundamental core assumptions that often are referred to in Mendelian randomization. There are other assumptions, but these three are pretty fundamental and important to understand. A battery of the tools in the toolkit that let us do Mendelian randomization pretty easily. And then with your humor permitting, I'll tell you a couple of vignettes of work that we've done in the last year or two. Some of it's unpublished, really using the toolkit to both make new discoveries and kind of clarify existing relationships. And then time permitting, I always like to gaze a little bit into the oracle, into the future, to see where I think things are going and directions I think are really opportune to think about. So let's turn to the intuition and the framework. And I think the nice thing about MR and science I like to do, I like to have a good intuition about it. And I think MR is one of these cases where it has a very nice analogy and intuition. And so here you can think about it analogized to the randomized control trial. So basically, it turns out nature, by the random process of mutation and spreading alleles as they've spread through the population through drift, of which some smaller subfraction actually perturb phenotypes, nature's kind of like done randomized control trials and we didn't even know it, where the gene is actually the perturbation rather than the drug or intervention that you would normally have in RTC. So if you think about the study design on the left in a randomized control trial, what might you do in a very simple case? Collect a population of individuals, split them into two equal groups that are ideally indistinguishable from each other for confounders of interest. You provide one of those groups a treatment. Let's say you give them a statin here, thinking about the relationship between cholesterol levels and cardiovascular disease risk. The control, you either have standard of care or some placebo or something like this. And then ideally, you sort of walk through one thing. Ideally, you monitor the groups. You see that the group that received the treatment had a change in the biomarker that the perturbation is designed to target. And then you monitor over time the impact of the change of that reduction of the biomarker and outcome of the event you're interested in this case, cardiovascular disease. And of course, we know that giving people statins lowers the LDL cholesterol, and LDL cholesterol is a causal risk factor for CVD. So of course, this randomized trial is great, perfect, and wonderful and ethical, okay? So now if you turn to the figure on the right, you can see the genotype. The design here looks very similar to the RCT, and the only real difference is that rather than the perturbation being a drug, in fact, the genetic variant that you've selected in the population that you know perturbs the biomarker of interest, so a genetic variant that influences LDL cholesterol, that's actually the perturbation analogous to the drug in the randomized controlled trial. Okay, so to kind of like really ground that in a very specific example, sort of like the favorite one that many of us like to cite is this variant at PCSK9, prohormone convertase 9. It turns out that many moons ago, seminal work by Cohen and Hobbs discovered loss of function variants segregating in this gene. And it turns out individuals that carried these loss of function variants had substantially reduced LDL cholesterol on the order of like 20 milligrams per deciliter, which is an amazing LDL cholesterol reduction. So then it sort of stands to reason, well, if these carriers, have lower LDL cholesterol and nothing else happening at the time, and it still seems vaguely to be the case that this is sort of like a tight perturbation on LDL cholesterol and that only, we can ask the question, do these carriers of this sort of like unfortunate mutation also have lower heart disease risk? Epidemiologically, you'd predict that if an individual had lower 21 milligrams per deciliter lower cholesterol levels, they should be protected against heart disease by about 25% or so. So now you can go to the observational association genetic data and ask that question. And it turns out, yes, in fact, they are protected, and protected proportional approximately to what you might have expected from the epidemiological case, that those carriers have a 20 to 30% lower rate of heart disease. So that's all great and interesting. When I sort of got into this foray, it sort of like emerged in this very interesting case that took us forever to get published. Really trying to understand not the role of bad cholesterol in the case that I just described with LDL cholesterol, but actually the role potentially of the so-called good cholesterol, high density lipoprotein cholesterol, and its role in cardiovascular disease. And for many of us here who obviously know, the development of HDL raising drugs as a therapeutic for heart disease has been this fraught pursuit. And at the time, there were like multiple clinical trials, billions of dollars had been spent. And none of those drugs had made it past efficacy trials in late stage randomized control trials. So great drugs to raise HDL, but they don't seem to do anything about heart disease risk. So at the time when we did these studies, we were really trying to understand, does genetic perturbation on HDL actually influence heart disease risk at all? Because if the genetics supported it, then it suggests that maybe actually there's something about the drugs or something about the perturbation we don't understand, but maybe genetics could give us clarity on that point. So in an analogous way to thinking about this with the PCSK9 coding loss of function mutation, you can think about strong perturbations on HDL. So we considered one particular genetic variant in endothelial lipase, a missense mutation that actually has a profound impact at HDL raising. So carriers of this low frequency variant actually have 6 milligrams per deciliter higher HDL levels. Epidemiologically, you would predict those individuals would actually also be protected from heart disease under the hypothesis that HDL actually causally relates to risk of myocardial infarction by about 10 to 15% or so. But now that we have a genetic instrument, we can actually ask that perturbation and see, do carriers of that variant actually have observably lower rates of heart disease. And the long and the short of it is not obviously so. Probably the most mileage I've ever gotten out of result with a p-value of basically 1. Here's the meta-analysis results accumulating about 21,000 MI cases with 95,000 controls across multiple case control and prospective cohorts. No evidence that carriage of this variant changes your risk of heart disease at all. And it's not from a lack of power. If you do the power calculations, we definitely have about 90% power to observe the epidemiological effect. So we can't rule out necessarily that there's like this tiny HDL effect that it protects you from heart disease, just not at the epidemiological expectation. But we can definitely rule out that it can't possibly be the epidemiological expectation with about a 90% confidence. But don't just take this one coding variant for its word. You can also, a reader would nicely point out that, you probably have multiple genetic perturbations for HDL and maybe for LDL too. What if you combine that information in aggregate? Do you also see a similar effect? And the answer is nicely yes. So if you aggregate at the time, the collection of known LDL biology, genetic elevation by those mediated genetic factors definitely increase risk of heart disease, even more than the epidemiological estimate. And we could have a discussion about why we might think that is, but definitely consistent with the idea that LDL is causal and genetic elevation increases your risk for heart disease. So perfectly consistent with everything we know about that biology. But combining all of the risk factors for HDL that we know about didn't seem to have any obvious association with heart disease risk at all, non-significant association. The expected effect on, should be about a 50% risk reduction. We're not even close to that. So not just true for an individual SNP, but an aggregate of SNPs in collection. And by and large, I think this basic result for HDL has stood up over time, though I think probably it stands to be reevaluated with the amount of genetic data that we have, which is substantially more than we had 10 years ago today. But I suspect probably is going to be consistent given the pharmacology that we know about, or the lack of success of those pharmacological perturbations on HDL. Okay, with that intuition and framework out of the way, I can't help but give a little bit of intuition about the basic model. And for those maybe a little bit sensitive to math allergies, this is your trigger warning that there's a little bit of math here, but don't worry, I'm with you, we're with it together. You'll be able to handle it, we'll be able to get through it. Okay, so really comes down to this kind of like mediating graph, and we think about sort of estimating different parameters on this graph. So if we start, looking at the left-hand side, you've got your genetic instrument called, capital G of J. So you've got your SNP, you've got your marker of interest, your biomarker of interest, capital M, and your exposure of interest, capital Y. And really what we'd like to infer is this parameter gamma, which is the relationship between the exposure and the outcome. Okay, so if we're armed with two measurements, which you can think of very intuitively. So if we knew the relationship between how change in genotype impacted change in the exposure, so basically just the beta effect sizes that we estimate from, genetic studies of, gene to marker exposure associations. And in a similar way, the beta relating the genetic variant to the outcome of interest, beta G to Y, the association between genotype and the outcome. You can kind of see how this is set up. You can simply calculate this very, very simple ratio, which is basically the change in outcome divided by the change in exposure. So if you were thinking about this, it's really just a slope in some ways. You're just performing a very, very simplistic regression. You're really interested in like, how does the change in exposure impact the change in outcome? And if gamma is 0, then you can say, oh, well, there doesn't seem to be any obvious relationship between genetic change in exposure and genetic change in outcome. So there doesn't obviously seem to be a causal effect. But if gamma was significantly different from 0, you might reject that simple null hypothesis. So you can think about this with a single genetic variant, but then of course, if you imagine just plotting all of those genetic variants, let's say you had a handful of them, you could plot a very simple plot here where you just plot the effect sizes of the exposure relative to the effect sizes on the outcome. And simply intuitively thought if you calculate the slope of that regression equation that tries to take into account that different SNPs have a little bit of different information. Some are more certain in terms of the relationship between genotype and outcome than others are. So if you weigh that variance accordingly, the slope of that regression is in fact that gamma that we're interested in, the change in exposure to the change in outcomes. So if we could estimate that gamma and do that with very standard regression or meta-analysis techniques, that's in some ways the basic way to do MR, and that's all there really is to it. So with that regression equation and with that basic math comes with some assumptions. So now what I'm going to turn to is just talking about a handful of those assumptions and what they mean. So in fundamental terms, there's three key assumptions that are required for this causal effect estimate gamma that we're interested in to be accurate and unbiased and robust. They are as follows. There's the relevance assumption, an assumption called exclusion restriction, and the independence assumption or the exchangeability assumption. And I'll walk through each one of these so that we can kind of make sure we understand all the terms and what we're getting ourselves into, okay? The relevance one is actually pretty straightforward. And so basically what that means is that, man, you better pick variants that relate to the exposure of interest, and they better be reasonably good instruments, right? So, and analogously, it's sort of like imagine like, in the case of a randomized controlled trial where you're imagining a drug treatment, right? You wouldn't design a clinical trial with a drug that was a pretty weak drug that only mildly did perturb the particular marker of interest, because if you did, you'd have extremely bad power to actually run the clinical trial in the first place, right? So analogously, what you want here are genetic instruments that are pretty potent. And so in practice, what does that mean? Well, what that means is that we tend to try to select genetic instruments into our kind of aggregated score that pass a basic genome-wide significance criteria, and ideally have been replicated in multiple studies. So they're kind of like the most robust of the robust tag variants for signals that we know relate to the exposure of interest. And the good news is that for a variety of traits of recent date, with the advent of sort of like DNA biobanks, we actually have quite a few number of the size of these genetic instruments is actually large. We can actually explain a non-trivial amount of variance in the exposure. So with a little bit of work, you can generally get pretty good instruments here. The issue is if you didn't do that, and this can happen sometimes if you're picking traits that are a little more bespoke or perhaps are newer traits, so haven't been profiled in large samples fully and completely, you can run into issues which the literature refers to as weak instrument bias. And what that effectively means is the bias in the effect size estimates when the perturbation is not a strong perturbation. And the more, the weaker your instrument is, the more potential for bias that you might incur. And that bias can either induce false positives, or even in the case where you've got a true association, will wildly misestimate the actual true causal effect that you might see. So you might ask, okay, well, weak instrument bias sounds bad, Ben. Like, what do we do? Okay, so typically what we do is two things. We'll first measure the amount of variance explained with the genetic instrument of interest. So if we're generally explaining a good proportion of the genetic heritability of the exposure, that probably means we're in a good shape. We've got a good, strong perturbation of that exposure. The other thing that we can do too is we can tabulate what's called an F statistic. And I've got some citations at the bottom. The F statistic is basically, it gives a measure of the in some ways vaguely the power of the instrument to make inference. And it's related to the phenotypic, the R squared, and maybe the size of the genetic instrument in fundamental terms. But the community generally, with some work from some seminal leaders in the field, basically have looked carefully at this. And for the most part, if you've got a genetic variant with that scores highly on one of these F statistics, generally you're in pretty good shape, and that the bias that if you did incur due to weak instruments is sufficiently small that you can probably not worry too much about this particular assumption. The second one is called exclusion restriction. And so this one's probably the most complicated of them, but it's intuitive and straightforward. So basically what you want in your MR experiment in truth is for the arrows of causality to point through the exposure and not anywhere else. So if the genetic variant somehow it was implicated somehow or associated or biologically generated an association to the outcome, not through the exposure, that can lead to some issues in the estimate of that causal effect, right? So, you know, the assumption here is that the genetic instruments that you're using, the effect that they're estimating on the outcome happens exclusively through the exposure. That's why we call it the exclusion restriction. If the exclusion restriction is violated, what could happen? All sorts of things can happen. The effect estimate you get can be biased. So if you imagined genetic variants that happen to associate on the outcome and sort of like elevated the effect sizes and you just didn't know it, you could get wildly overestimated effect sizes from exposure to outcome. Depending on the directionality of those bias of that sort of like extra step, you could induce false positive associations or even false negative associations too. And in principle, if you thought about it, you probably can recognize that this really relates to what's called horizontal pleiotropy. So basically, you've got additional trait associations that you haven't accounted for in this very, very simple causal inference graph model. And so in practice, what the community does is basically, you know, generates methods and applies sensitivity analyses that we think are robust to violations from this assumption. And if we maintain consistent causal effect estimates, we're generally reasonably happy that if the exclusion restriction is violated, it's not violating it enough sufficiently to change the inference of our experiment. The final assumption is that of independence or exchangeability, and this one's also pretty intuitive. So basically, we also want the instruments and the genetic variants that we choose not to be associated with other confounders outside of the exposure. So for example, you know, an intuitive one that I often think about is, you know, if our exposure was body mass index and we wanted to relate body mass index to cardiovascular disease or type 2 diabetes, we know body mass index and waist-hip ratio are correlated, and we might suspect waist-hip ratio might also impact diabetes risk too. And genetic variants definitely relate to both waist-hip ratio and BMI. So that might influence our inference of a causal effect between BMI and an outcome in that way. So much like the exclusion restriction assumption, if you've got issues there, it can bias your causal effect estimates depending on the directionality of the confounder's effects on the outcome. You can, of course, generate false positives, but also false negatives if they sort of masked exposure effects. So practically, what do you do? Well, if you happen to be in the nice case where you actually know about the confounders of interest, then there actually exist methods that we'll talk about that allow you to control for those effects and then create causal effect estimates that regress out or adjust out the effects of those confounders, just like you might do in a instrumental variable regression where you have the direct genotype data. So you can definitely do that. If in the other circumstance you didn't know about trait pleiotropy, like there might exist confounders like hidden you know, creatures out there that were trying to just give your experiments a bad time and you just didn't know what they were. Well, one tactic we often do is to consider what additional associations our genetic instrument might relate to, like consider the pleiotropy of our instrument and make sure that the traits that that instrument might associate with aren't obvious confounders. So do something a little more exploratory. And if we did discover something we thought might be relevant, then apply existing tools to try to take care of that. That's what we do. So it turns out that dealing with these assumptions and developing new methods to try to cope with them is an active as you can imagine, a highly active area of investigation for the biostatistics community, particularly at Bristol and Oxford and Cambridge. So I'll go through and talk about a couple of tools that I routinely use, and this is by no means a way of being comprehensive. I'll point you to some links at the end to give you a location to find sort of like the complete battery of all the things you could do. But I'll tell you about the couple of things that I use in my work that I think capture like 90% of what you kind of want to look at here. So the primary statistic that generally gets used is what's called this inverse variance weighted estimation procedure. And again, it's a long-winded way of describing that regression equation that I described above, where basically every SNP has an effect, and the effect that you have on the exposure or the outcome has some variance. And so you want to combine all of those together to estimate that slope. And you want to weigh it by the amount of information that each of those particular genetic instruments offers. And that information is proportional to the inverse of the variance. So just in an analogous way that we might imagine a meta-analysis based on a fixed effects model or random effects model, the inverse variance weighted methods are exactly analogous to those procedures. The field generally prefers the random effects model to the fixed effects model. And I've kind of stated the fixed effect model assumptions here. And you can kind of see that actually, it may be true that every genetic variant gives you a single direct estimate of the causal effect, but that's a pretty strong assumption. And so actually, just for conservativeness, the field tends to prefer random effects models where maybe there's a little bit of uncertainty SNP to SNP about its relationship in inferring the causal effect estimate. So it includes some information about heterogeneity when it does the inverse variance weighting. And this is principally like the first and predominant model that people utilize. It's the most sensitive to violations from the MR, the Mendelian randomization assumptions that we've described, but it's also probably, as a result, not surprisingly, the most powerful of the approaches that are described. One method I think is pretty handy, even though it sort of has the least power of all of them, is the MR-Egger approach. And this particular method is actually robust to that directional, that exclusion restriction assumption in the following way. So if it turned out that in your genetic instrument that you built, let's say that the instruments that you selected were invalid because they had some exclusion restrictions. There was some additional pathway to the outcome that was caused, so some level of horizontal pleiotropy. But let's imagine that horizontal pleiotropy was always in the same direction. So let's say the allele that increased the exposure that you were actually interested in also somehow increased the level of some other hidden exposure that you didn't know about, but they all did it in a directionally consistent manner. So what might you see? So if you look at the graphs here, in the case where you didn't have this happening, you'd see something on the left where you sort of had this nice relationship between exposure and outcome. But in the case where you have this directionally biased horizontal pleiotropy, what you might see is the graph on the right, where actually the slope of the line is not that different from what you would see on the left, but actually there's this bias that's added where the intercept no longer crosses zero, it actually crosses some elevated level, let's call that intercept term alpha. Okay, well, we're in regression space, so the game that MR-Egger basically plays is, well, let's actually just assume that that's true, that there's directional horizontal bias, and test that assumption and try to estimate that intercept term alpha. And if alpha turned out to be non-zero, then we can conclude that there may be directional bias due to horizontal pleiotropy. And then we can re-estimate the causal effect just as we did before, but just including the intercept term correcting for that bias when we're doing our regression. And it turns out that that generates a valid causal effect estimate, gamma, in the context of this directional bias. So there's a tool that does this, and typically we use MR-Egger and the estimate of that intercept term to determine if there in fact exists bias, and if there is, adjust our causal effect estimates accordingly. There's another version of the MR estimation called the median estimator. So, so it turns out, and if you're following kind of like the statistical intuition at home, this will be very intuitively obvious. Okay, so we kind of have this idea about estimating quantities based on the mean or estimating them based on the median. And it turns out, right, like in the presence of outliers that are extreme, the mean tends to be a little bit unstable, whereas medians tend to be pretty robustly stable. So the intuition here is that actually, well, what if it turned out that in the collection of all the genetic instruments that you had, most of them were actually valid instruments, or most of the information you derived from the causal effect estimate came from the genetic variants that were actually valid. So they met all the MR assumptions, but you kind of had a couple of bad apples, or the minority of information that you might infer comes from instruments which may not meet entirely all the assumptions of MR. So if it turned out that that was the case, then what you could do is actually construct a median-based estimator based on a random effects model, rather than the mean-based estimator based on the inverse variance weighted random effects approach. And it turns out that in the case where the majority of the information comes from valid instruments, or in this case, the minority of information comes from invalid estimates, then that median-based procedure actually returns a calibrated, robust estimate of the true causal effect in that case. It's kind of a little bit of stats magic, but again, intuitively, it just comes with this idea that estimates based on the median tend to be more robust to these kind of like extreme outliers, and you can adjust your estimates accordingly in that way. So it turns out actually all of these methods are implemented. So it's not like you have to do like the statisticians that create these things, embed them in packages. And it turned out, like I said, in about around 2018, a lot of these tools got codified in this toolkit online called MR-based that implements not just these methods, but a catalog of additional methods, a bunch of plots, and a bunch of other data. And it just really democratized the use of this approach, because it just streamlined a lot of the procedures that we use to actually do the MR experiment in the pipeline itself. And so just to give you an idea of what that pipeline looks like, this comes from Hamani et al. In 2018, where they kind of describe what MR-based is doing behind the scenes. But just to give you an intuition about it, right, what you've got to do is you've got this kind of like two sample MR design, where you've got a genome-wide association study for your exposure of interest. And from that, you want to kind of choose the genetic instruments, tagged variants that map to the signals that of association that you found from that study, selecting maybe one sentinel genetic variant from each signal, perhaps. And then you go to your outcome GWAS of interest, and you really would like to just look up those genetic variants in the outcome GWAS to get those associations between the genetic variant and the outcome from those GWASs. If you had those genetic variants exactly, you could just read them out, but maybe you might need to get handle linkage disequilibrium, look for proxies for those things. And you certainly need to harmonize the data too. So you want to make sure that the alleles that you're selecting for the variants you pick are the same alleles. This can be a trouble if you're running into so-called palindromic variants, where it's a CG or an AT SNP. And you want to make sure that the exposure GWAS, when it refers to a C allele in a given variant, that the outcome GWAS is also referring to the C allele, and that's the C allele in the positive strand in one and the C allele in the positive strand in the other. So it helps automate and homogenize that process to make it very convenient. And then once you have that procedure in place, you can generate all of these causal effect estimates, sensitivity analyses, and plots of merit to go about your way interpreting and leaving you, the user, to actually interpret and understand the data rather than have to get caught up in building the pipelines and executing them. One other tool that I think is also pretty important here thinks about trying to deal with that confounding assumption, right? So as I kind of alluded to earlier, it's not unreasonable to imagine cases where you're imagining, okay, I want to understand whether or not, in this case, waist-hip ratio is a causal risk factor for type 2, but I know any genetic variant that I picked that I knew was associated with waist-hip ratio is almost surely associated with body mass index. And so with the conventional arsenal of univariable MR tools, I would be stuck and I'd be horribly confounded because I couldn't deconvolve the effects of body mass index and waist-hip ratio to estimate that question mark relationship between waist-hip ratio and T2D. But it turns out that actually there are tools, multivariable tools which allow us to disentangle that confounding. And intuitively, you can see how that might go about, right? So if for genetic variant, we have the relationship between it and each of the exposures of interest, the exposure we're keen on, and then the confounder effects, then we can imagine a regression equation that just basically, in some ways, like regresses out the effects that we're less interested in. So the genetic effects between body mass index and T2D, and then anything that's left over is the effect of waist-hip ratio on T2D, and that process and procedure, again, under the usual MR assumptions and maybe a couple of additional ones, returns an effect estimate between the exposure and the outcome given these additional confounders. So that's another tool in our toolkit to help us understand and deconvolve potential confounding effects from causal risk factors when we're interested in specific ones that we know are correlated with them. And I don't have, like there's tons of things to think about in building a great genetic instrument and kind of like calibrating accordingly. I've kind of got a short list of some of those tools that deal with either outlier detection for your genetic instrument that take advantage of all of the tools and gives you sort of like an expert meta-analyzed average Bayesian model averaged guess of what the causal effect is, assessments of directionality or other methods. Tons of work in this space, tons of methodology that exists. I found this handy website actually by our friends and colleagues at Bristol who have thought a lot about this. That if you're all interested and just want to make sure you understand the terminology and explain well, that's a great place to go to kind of get up to date on these things. Okay, with that kind of background in place, let me turn to some vignettes that we've worked through in the lab, and I'll kind of walk through a handful of them. The first one I'll describe came out of work from a postdoc who's now an assistant professor at the Children's Hospital Philadelphia and a master's student who's now a grad student at Penn, motivated by what we thought was a very straightforward finding, but then it turned out to be complicated. So really the question was, does smoking behavior influence susceptibility to type 2 diabetes? Okay, so it turns out there are large-scale genetic studies relating smoking behavioral phenotypes that exist, and they're pretty large and robust. And so it's possible to build genetic instruments for different types of smoking behavior. In this case, we selected smoking initiation for better or for worse. So it turns out that at a first pass level, a genetic instrument developed from the largest smoking initiation study that we had available included 341 SNPs, and using kind of like the battery of the methods I've just described, inverse variance weighted, weighted median, and MR agar, seemed to suggest that elevated genetic elevation in increased smoking behavior increased susceptibility to type 2 diabetes, which may at first blush not necessarily be a surprise because we all know that smoking behavior definitely is a causal risk factor for heart attack. And that's evidenced by the plots below, where we can take the same genetic instrument and GWASs for coronary artery disease and see a very consistent positive association between smoking initiation behavior and risk to CAD. So this all looked very promising and compelling until we started to explore this a little bit in more detail and realized that actually our smoking genetic instrument was also positively associated with strangely higher BMI, which we don't totally understand, even though there's some anecdotal support for the idea of a relationship between BMI and smoking behavior in this directionality, okay? So because we also know that BMI is a risk factor for type 2 diabetes, it just stood to be reason of like, oh, well, actually, could the observed relationship that we see in this univariable case just be explained by BMI, okay? So we take out our multivariable Mendelian randomization toolkit and reassess our relationship. So we ask, does change in smoking behavior in the context after adjusting for BMI increase risk to type 2 diabetes? And the answer is basically the result is massively attenuated. So the odds ratio goes down massively. The p-value is like barely significant, 0.03, so technically rejecting the null, though pretty close to the null. But in contrast, CAD definitely still has a persistent effect. So our positive control here isn't obviously explained by BMI, and that makes sense because we know CAD, smoking behavior has non-BMI effects on heart health. So that all made sense. But we were a little bit dissatisfied by this, and really we would like replication to kind of like bear this out. So what we did was we turned to the Million Veterans Program. So it turns out the Million Veterans Program data is this sort of like genetic resource that has hundreds of thousands of people from diverse ancestries with all the phenotypes that we're interested in evaluated. And so we actually have smoking behavior, BMI, and T2D all conveniently measured in the same set of individuals. And it turns out you can definitely do instrumental variable analysis in the context of one sample data where you have all these things measured. So we basically redid this experiment in those data. And it turns out that we can find basically everything that we saw before. So can we, when we ask the question, does smoking behavior correlate with increased BMI? The answer is yes, it does. That's exactly what we saw in the 2-sample case. Does smoking behavior on its own in a univariable model increase susceptibility to type 2 diabetes and CAD? Definitely yes, we see both of those associations. In the context of BMI, does smoking behavior increase risk to CAD? Absolutely, definitely consistent, positively associated, just like we did in the 2-sample case. But then when we look at type 2 diabetes, given BMI, does smoking behavior influence susceptibility? There, we do not see a positive association between CAD or T2D and BMI. I think unfortunately, I projected the wrong slide. I copied the wrong thing, but this comes from a paper. The p-value is basically the null hypothesis. So we can't, maybe there's a small effect that's not mediated through BMI, but all of this seems to suggest that if there's an effect of smoking behavior on T2D, it's a pretty minor effect, if at all. The second story I'll tell you about is a news story that's unpublished about adiposity and blood traits. And again, this is work led by Chris Tom, who's a pediatrician interested in blood cell biology. And for those that don't know, I don't know a ton about blood cell biology, but I do know that a lot of work has been done. And it turns out on the developmental biology of blood cell biology, a lot of work is known. And we know that blood cells, red blood cells, platelets, and white blood cells are created by this, developmental process stemming from hematopoietic progenitor cells that differentiate into different types of cells that ultimately lead to the diverse population of cells that do all the things that they do in our body. How those cells differentiate involve both extrinsic mechanisms, so perhaps the microenvironment by which those cells are differentiated, but also autonomous mechanisms, the catalog of transcription factors that are expressed at key times in the developmental biological milieu that lead to these different cell types at key times, at key places. Focusing on the extrinsic mechanisms, in particular, bone marrow is actually a pretty complicated tissue. Your blood cells are made in bone marrow, and if you look deeply into what's actually in bone marrow, you'll see that the different types of tissues that are both intrinsic to the bone, so osteoclasts, osteoblasts, and others, as well as other immune biology cell types, and it turns out adipose tissues are all present in this tissue. And so when we were thinking about this, we sort of thought, well, there's this sort of like increasing amount of literature that is sort of suggesting that actually that cell type population, the constitution of what's in the bone marrow, have intrinsic properties and influence what types of blood cells are actually made. So, and all this has kind of come out, pretty recently in the last couple of years. And in particular, thinking about the adipose obesity line, it turns out that obesity-related changes might actually influence that microenvironment and perhaps drive either cell expansion, influence cardiovascular disease risk, maybe promote clonal hematopoiesis and HSC stem cell progenitors, or even the fates of the ultimate cells that are produced. So the appreciation of adipose tissue in bone marrow as a contributor to what types of cells are made and how many are made is not something that has been fully appreciated, though has recently gotten new attention. And, I'm a human geneticist, and I don't often think about sort of like the detailed, deep mechanisms of blood cell and adipose relationships, but I definitely think about physiology. And we know that actually the distribution of weight and where different adipose tissues are stored in the body are both genetically influenced traits. And there have been a large number of genetic studies that have dissected both of these traits deeply. And sort of what we kind of understand about body mass index, for example, is sort of like its relationship to insulin sensitivity and the body recognizing the signals of satiety, so implicating neurobiology. Whereas measurements of where the body stores fat is quantified by waist-hip ratio, seems to implicate adipose functionality. So dysfunction leads to different places where that fat is stored when you intake a lot of it. And so given these physiologies, Chris thought, well, have we actually understood if any of these physiological measures actually map to blood cell traits? And there's some evidence that suggests that body mass index sort of, morbidly obese people have anemia. So there's some epidemiological reports in the literature, but nobody's ever tried to correlate genetic change in any of these traits with blood traits. So he embarked upon that and finds the following. So I've got sort of like in these plots, just as we had before, these are causal effect estimates relating either one or two of these traits. In this case, on the left, hemoglobin measures, and on the right, hematocrit measures, two different types of measurements of blood traits. What you can see in this sort of like univariable case in the top row, change in BMI, actually higher BMI lowers hemoglobin and hematocrit, whereas elevated waist-hip ratio actually raises hematocrit and hemoglobin levels. So these two traits seem to paradoxically have opposite effects on blood traits. And you can even further see that effect deconvolved when you perform the multivariable experiments where you build instruments for waist-hip ratio or BMI and adjust for the other trait. You can see both of these traits also consistent where BMI effects are lower, higher BMI lowers blood traits, whereas higher waist-hip ratio actually raises both traits. He thought maybe, it's interesting to see this in blood traits, but then he asked, well, what about other types of cell populations that are also created in bone marrow? We know that platelets and white blood cells are also created there. So he applied his those same genetic instruments for BMI and waist-hip ratio to these other traits. And what he observed was basically consistency of these observations across those different cell populations. So higher BMI lowers platelets, higher waist-hip ratio raises platelet counts, higher BMI lowers white blood cell counts, higher waist-hip ratio raises white blood cell counts. So pretty consistently across these cell populations. And what biologically taken collectively, this sort of suggested to him is actually that there's something multi-lineage going on. A simple model might be a perturbation in sort of the cell populations that are the progenitors of each of these populations. If they were perturbed in a way that's sort of, where somehow genetic elevation and BMI sort of like perturbed that to repress differentiation of those cell types, you might see this effect, whereas waist-hip ratio and a constituent of change in adipose density or distribution might actually influence blood traits in the opposite direction. But it seems like a simple model might be targeting that one cell type population with either of these two physiologies might matter. So suggest something multi-lineage, not something specific to the lineage. So it kind of suggests that, in the conventional model where maybe chronic disease leads to anemia, maybe actually there's this sort of progenitor cell type that might be influencing these traits, where this extrinsic factor, the physiology that you read out, had these opposing effects to balance out somehow the cell populations that you need to do normal functions in the body. I think I'll skip this story for the sake of time, and sort of like end here thinking a little bit about I think some directionalities for this experiment to go and some opportunities and prospects here. One particular area that I think is kind of exciting from the point of view of therapeutics, and you probably could intuit this given some of the examples that I selected with coding variants and genes that are presumably druggable targets, is the idea that maybe what you could do is actually select genetic variants that mimic perturbations on a gene that's druggable, and estimate or quantify the effect of those perturbations on exposures and then maybe outcomes, with the idea being, well, if you take the example on the right-hand side, well, what if I handed you genetic variants that we knew changed the expression or influenced the function of HMG co-reductase, the targets of statins? The prediction would be that if you perturb and reduce the function of HMG co-reductase, or maybe the expression of HMG co-reductase, you'll lower LDL cholesterol and perhaps lower heart disease risk, because that's what statins do. And so here's a collection of genetic variants that are selected for exactly that, and measured as sort of like the effect these variants have on the effect on coronary heart disease. And you can see that by and large, many of these have non-trivial effects on heart disease risk taken in aggregate. So, and this is just like one very toy example, but you could imagine doing this with a range of other targets putatively of drugs. If you've, again, got solid perturbations that you think definitely impact the gene and maybe don't impact other genes, you could imagine saying, oh, this is a druggable target. If I drugged it, what would be the phenotypic consequences of that perturbation? You could use the Mendelian randomization framework to kind of make that inference. I think this is a particularly nice idea because with the availability and the recent publication of large-scale protein quantitative trait locus genetic experiments, where basically, Decode published this massive paper with like 5 terabytes of data where they've quantified in thousands of people genetic variants that influence the protein level expression of thousands of circulating protein biomarkers. So if you went to that resource and you found variants that influence the protein expression of those genes, you could use those as genetic instruments to ask, well, do those variants perturb or influence an exposure that I think is causal for an outcome? Then that might give you evidence that that biomarker perturbation might be a reasonable target to go after druggably. I think there are a couple of other areas that are kind of interesting that I being a member of the Million Veterans Program data, sort of like calibrates me towards. I sort of described a couple of mediation experiments that I think are interesting. I think there's a lot of room to grow in interpreting the causal effect estimates that we kind of take for granted by considering pleiotropy. I think that's one area that is really exciting and I think is worth doing. Taking advantage of studies where you have all of the measurements in the same cohort and you can adjust for additional confounders that you've measured phenotypically or clinically, I think is also extremely exciting. So biobanks like UK Biobank or Million Veterans are great examples where you could do those experiments really nicely. It goes without saying, and it's horribly disappointing, that all of the studies that we have collected large-scale human genetics data on, by and large, are not as diverse as they ought to be. But increasingly, genetic surveys of diverse ancestries are becoming available. And so we can reapproach all of our causal inference questions in the context of those diverse ancestries and make inferences of causality in populations that are definitely underserved and would be best served by having those causal insights. I think that's incredibly exciting. And finally, thinking about traits of progression. So we think about our traits as endpoints like diabetes or BMI at a single time point, but we know BMI, blood pressure, LDL cholesterol, all of these things develop over time. And so if we had phenotypes that read out that progression, that would give us incredible resolution to understand the relationship to the outcome and how changes at a much more higher resolution would influence that outcome. I think all of that is definitely possible and things I think we're going to start to see in the next couple of years. So with that, hopefully I've told you a little bit about how human genetics data can provide evidence of causality, and maybe even just inspired you to go investigate some of the tools out there to ask your own questions, and giving you a couple of examples of places where we both can kind of clarify existing effects and find evidence of new causal effects that may point to new biology that we haven't fully appreciated. You've been a wonderful crowd. Thanks for your attention. I didn't do any of it. All of my great people did, and my collaborators helped out a lot. Happy to answer questions, and thanks for listening.
Thank you very much for the great talk. I would like to open this for discussion. If anybody has questions, just unmute yourself.
Hey, Ben, great job, and the RITS really appreciates your time and efforts. That was incredibly clear. I was wondering, and certainly Dan Rader and I go way back, when you start talking about, the inferences and the association, have you seen in your space at Penn evidence that they've taken the data and moved it into an RCT or directly had an intervention that could modulate the response that would be a feedback and confirmation of the value of the approach? You talked a bit euphorially about predictive, sort of pharmacoeconomics around, or pharmacodynamics. I was just curious.
Yeah, that's a tricky question. I know that I think probably the places where that is really trying to be thought of in the context of what Dan Reeder thinks about is lipids and lipid subfractions, because I think he thinks that there's more biology that we don't yet understand around HDL and reverse cholesterol transport that, if we could clarify more precisely, would lead to targets that are of interest. So I know he's thinking about that. I mean, I think it's early days in that space, so nothing immediately obvious. And then the only other place that I know, I mean, it's close to my home again. My biases are like cardiometabolic and the cardiometabolic disease space, but liver traits, in particular, fatty liver traits. I know that that work has been stymied, I think, a bit by the lack of robust GWASs for fatty liver disease, because it's just a really hard trait to phenotype. And so, the place where we've begun is basically to collect data and do phenotyping better, either by proxy or with imaging that's available in biobanks. And then once you have that, you can do the genetic studies and then do the discovery and then start to motivate the trials to kind of like chase down targets or biomarkers you think are of interest. So I think my understanding, I think these are very early days, but so still promise more than anything. Like, I think you'd have, like, drug companies are the place where I think I know for sure this is really being picked up. Like Regeneron and monoclonal antibodies to PCSK9 is probably my most concrete example. And I think I get headhunted all the time for people asking me to be statistical genetics and apply MR so that they can scan UK Biobank data better. So I think this is really active in the private sector for sure.
Yeah, there's no doubt they're talking about in silico RCTs using these data combined with an EMR, right? Which is intriguing. And one other follow-on question, when you start to talk about the SNPs, it smacks a little bit of candidate gene analysis where you already know what you're thinking of looking at. It's not an unbiased view. Could you imagine in some way, shape, or form in the future that you would have an unbiased approach for discovery? Because what you're asking is, I have a gene and I want to know if the outcome and exposure occurs, but I have to have known that that gene may have value, you see?
Yeah, and so I'd answer that in two ways. Like, in practice, I think what you routinely do is you actually think about this in an unbiased way. So if you thought, like, LDL cholesterol was a risk factor for type 2 diabetes, what would you do? You would go to the latest and greatest global lipids genetic consortium, GWAS, select sentinel variants across the genome. I wouldn't care what the genes are necessarily. Build a genetic instrument and then sort of like curate that instrument to handle pleiotropy and other sort of like confounders. So intrinsically, that's the mode that we operate on. But we do the kind of candidate gene kind of experiments if we're very keenly interested in a gene because we think it's druggable or we're keen on it. So there's definitely these kinds of other experiments that are like targeted in that way. It's the framework's versatile. If you've got good instruments, you can kind of do it both ways, but we do the unbiased thing primarily, I think.
Great, thank you so much.
Ben, this is Bill Welsh. A really fantastic talk. I enjoyed it immensely. I'm so glad I tuned in to your talk. We just had a department meeting and I was going to pass, but I'm so happy that I was here for your talk. Thank you very much. But I do have a question about the relationship or which is the cause and which is the effect, and maybe reversal. And so it's you're inferring the cause effect relationship. But if we take the smoking induces higher BMI take that as a sort of physiological example that we're all familiar with, maybe. So I've noticed in my wife and my family, particularly my wife's family, the smokers tend to be more overweight, which is sort of. But my psychological profile of these people is that they gain weight and then they adopt smoking, because smoking and nicotine is fairly well known, or at least highly suspected, to reduce your appetite through cerebral type effects. So of course it's a treadmill effect, or it's a losing effect. So essentially, the study that you did where smoking induced higher BMI, maybe it was higher BMI induced incipient smoking. So if I take that to the next level, to the genotype phenotype, so maybe I'm wondering if higher protein, translational protein, production of protein, can induce higher gene expression. So essentially what you're looking at the gene causing higher protein expression, but maybe as the protein expression becomes higher, it's a feedback effect, and it induces greater gene expression. So production of the gene and then gene expression. So essentially you have cyclic inverse cyclic effect going on, and it's going both ways. So you have an equilibrium effect. So I just want your comments on that, and I'll leave it there.
Yeah, no, thanks for both of those. I'm glad you appreciate you tuning in, and I'm glad you enjoyed it. Particularly with behavioral traits, as you point out, the interpretations can easily be very complex, and they can feed back on each other. And so almost surely in the cases that we have here, we must be violating some number of assumptions that kind of go into it. What we're at least modestly reasonably happy with is the idea of trying to deconvolve the relationship to T2D, right? Because it would be incredibly, obviously clinicians should tell people to stop smoking, right? But then to say, oh, you should stop smoking because it'll also lower your diabetes risk, right? Like that would be an incredibly potent message if we thought that there was evidence to support it. But it could just be as simple as you nicely point out, that like maybe it's people are attracted to smoking because they're they're genetically predisposed to be heavier, and then that genetic liability increases the risk of diabetes. So it doesn't, like the smoking effect doesn't ameliorate necessarily their diabetes risk. If it does, it only does it through their BMI lowering, right? So again, like the messaging and the interpretation of these things is incredibly challenging, but just having the ability to have data to even like start to ask these questions and see what it means is, I think you're nicely point out is really important. On the biological mechanistic side about sort of like protein expression and transcription, this is another place where I think MR is sort of like starting to imagine expanding how it thinks about things. So the field has been driven in the methods it's developed by biostatisticians who know a bit about biology and how complicated regulatory biology is, both on the level of transcription and translation for sure. They appreciate it, but they don't, the models that we built don't really think about that kind of level of mechanistic detail necessarily. With the right kind of data, I think you could definitely start to build much more higher resolution graphs that start to think about the relationship between a genetic variant, how it's transcribed, how it's translated, and then how that impacts the exposure you're interested in, and then ultimately the outcome. It's not inconceivable that we will have all of the data for all of those edges in wide abundance someday, and we'll be able to kind of think about exactly the kind of feedback loops that may happen locally in a gene, or maybe as a broader biological mechanism that helps us do this interpretation. So very astute, very reasonable, early days. We're waiting for the data for them.
Thanks, man.
So Ben, is this something like a reverse causation? Doesn't MR correct for the reverse causation?
Yeah, that's one of the advantages of thinking about at least reverse causality on the exposure to outcome, because we think that genotype precedes phenotype. So, there is a natural ordering to that directionality. Now, there's some, you could still get a little bit confused on the directionality of exposures. And there are some methods that try to assess that by directionality too, because you can imagine cases where, violations of the assumptions that you make where the arrow of directionality is one and not the other way, where there's mediating effects that happen in different people that could cause differences in the exposure outcome relationships. And so some of those things are testable and some of them are evaluatable for sure.
And you touch base on the ancestry population studies. And we all like to have our science to be open, but some do not share their summary statistics. And how do you go about? Do you just contact the investigator and ask for the data set? And what have you encountered over the years?
Oh my God! Do you have a drink? I could tell you all the stories. It's really frustrating and it's really complicated. There are great people and great teams that basically put all their stuff on public domain and make you click a box that says, please don't identify these subjects. And that's what they do. And you can get access to the data immediately. And it's high quality and sufficiently resolute that you can do all the things that you want. To groups who just flatly refuse to release any of their summary statistics at all, even though they're publishing in open access journals and all of these sorts of things. It's frustrating. My current frustration, the stone that I have in my shoe right now is actually around MVP and the accessibility of those data, for exactly this ancestry reason, which is that Million Veteran Program data is like, probably 1 million people are ultimately going to get genotype, and it will include African Americans and Hispanic, Latinx individual ancestries, and that will be like 200,000 people, right? It'll be an amazingly large data set. So it's like something that, like, I think if it were summary data were in the public domain, it would be devoured ravenously by a community hungry to utilize the data to make inferences these groups. But the way that it kind of goes is it's like gated through dbGaP access, which you can get access to. So it's not like it's a barrier, it's not like it's inaccessible, but it provides a barrier to access, which is kind of challenging and frustrating. But at the same time, it's like the VA has its rules. It's like all these places that have restrictions have rules. They signed IRBs. They don't want to upset the standard practice. And so, just trying to work with people, like emailing them and asking them is my first line of defense. Trying to get access to the data and collaborate if possible. Usually works pretty well. And then just like to eat the administrative cost when you have to get access to dbGaP and other places. I feel like one should like hire somebody to do this as a full-time job, because if you did it, you could get the data, but you spend like hours toiling over it, which is unfortunate, but it's the state of the art. I wish it were better. The places that I really hate are like 23andMe. This is the place that just drives me crazy, where they won't release data, their own summary data. They will publish papers in amazing journals, but they will only release the top 10,000 SNPs from their scans. And if your institution doesn't want to sign the IP or material transfer agreement with 23andMe, which comes with these like incredibly protective IP requirements that they have. For example, Penn won't sign the 23andMe agreement because we don't want to sign the material transfer. We can't get access to the summary data even if we wanted to. So you see it all, and it's annoying and a headache. And I wish I had good solutions to it, except for every study that I try to make as much of that available as possible. I feel like it's a drop in the bucket, but if we all did it, I think we'd be all better off.
Any other questions? Well, thanks, Ben, for coming in on very short notice. That was very educational. Could you actually stay a little bit after, and we can probably chat on this soon?
Yeah, absolutely. No problem.
All right. Thank you, everybody.
Thank you, Stephen. Thanks for a great talk.
Thanks, Stephen. Thanks, Ben, again. Goodbye now.
So Ben, my wife is a statistical geneticist. So she
Oh,
trained
yeah.
with Steve Finch from Stony Brook.
Awesome.
And she then went into Penn State for a while as a faculty, and then she transitioned to National Institute of Health. So she's now at the Human Genome Institute, and she actually started this Mendelian. She got interested in it, trying to correlate the brain imaging and using the database, heavy database on the imaging to some of the neurodevelopmental disease and related traits. So when she was doing that, I'm like, oh, can I use this and actually look at some of the observational studies that have shown some, association between asthma and some related traits such as GERD. And we actually found some very interesting result between the GERD, gastro efflux disease, and the asthma. I think it has some, clinical relevance, because I think, how we treat asthma patient with GERD is kind of a mixed signal, because we try to give them, anti-proton pump drugs, but they don't pan out as a clinically effective. And our data seem to show that basically, very weak association through the MR. But interestingly, we also try to generate instrumental variable for eczema, which has been a kind of common etiology with asthma. And when we do that, I mean, it just gives a little more complexity to this whole story. So I was wondering, maybe I can send you the paper. Have you on board with this project and get your thoughts on it?
Yeah, sure.
Am I missing anything? Are we missing anything in terms of analysis? So I looked at your presentation and we check all the boxes that you presented in terms of all the sensitivity analysis and so on. But maybe there's something that we're missing. Maybe we would like to get your expertise on those.
Yeah, no, that sounds great. Send me the paper. Happy to do it. Yeah, I mean, the only thing as a human geneticist that thinks about pleiotropy a lot, I guess the only thing occurs to me just like gunslinging a little bit would be if there are other confounders that I know that are related to asthma and GERD. So like off the top of my head, I would, like, BMI might be one that I wouldn't be surprised probably is correlated with both of them. Right. And then, but thinking about traits like that, it's trivial to do, if you haven't done it already, to kind of like do the MVMR kinds of experiments to deconvolve those things, if that was a concern. But yeah, I'll take a look at, if you send me what you got, I'll definitely take a look and kind of like ruminate and see what makes sense. So.
Oh, that'd be awesome.
What's the directionality of it? Is it which way does the arrows go? Is it GERD to asthma?
I think it's asthma to GERD.
Asthma to GERD. Okay, interesting.
No. GERD to asthma is stronger. Yeah.
GERD to asthma. Okay.
Yeah.
So do you and what like physiologically, what do you think the biology is? What is it sort of like inflammation, and then sort of like you kind of just trigger?
Yeah, so I think that there are a lot of mechanisms that we haven't thought about. I mean, obviously there exists this reflux and reflex mechanism that may contribute to decreasing pH in the airway, that contribute to airway narrowing in asthma. Those mechanisms may work or are may apply to what our results suggest. But interestingly, those mechanisms can also attribute to eczema, the atopic dermatitis, which people really don't have thought about in those associations. And so, but atopic dermatitis does not cause GERD. There's no directionality in that direction, but there's a reverse direction where GERD can equally contribute to asthma. The effect size is pretty similar for GERD-inducing asthma and GERD-inducing eczema. So that was kind of interesting.
Is there what kind of epi exists? Is there like, has this been observed observationally before, or?
Yeah, I think there's a numerous epidemiological or clinical studies have shown asthma and GERD. I don't think there has been too much on the atopic dermatitis and GERD. And there's strong genetic association between eczema and asthma.
Yeah.
So yeah.
What's the? I haven't followed GERD genetics. Like, what's the biggest GWAS for GERD? That's interesting.
So there's a recently published paper that we generated instrument variable from. So I can, the manuscript entails 3 different independent studies, but some include as you said, 23andMe data set, but they don't share that, so we took that out as a generating a variable, an instrument ID. But yeah, so there are 2 recently published or in press data set. I think it's a meta data set, GERD, and there's one for atopic dermatitis. So that's what we use.
Okay.
And then obviously for asthma, we use the largest GWAS on the UK Biobank. Asthma definition.
Yeah. No, that's send the paper. I'll definitely take a look. I'll think about other data too. Getting, like, I doubt that the MVP has done a GWAS of GERD before. I would be surprised if there isn't an asthma study underway. And so, I just, that, down the line, I know down the line with the right seeds, like me asking asking the right people if something's been done might actually help to generate other summary data that then you could add to it over a longer period of time. So I'll definitely think about that too.
As you can tell, I'm not a geneticist. I just got briefly introduced to this new method.
If that's not true, you do an extremely good job of describing things. So you're in good shape. I wouldn't have guessed otherwise.
Well, I shared. We're generating the plots right now, the results, but I can give you in the form right now. It doesn't include any figures. Would that be okay?
Yeah, sure. Yeah, I mean, tables probably. Tables for like the primary MR results probably. Yeah, that's fine. Yeah, I mean, forest plots are always great, but I can read table too.
Good. All right. Sounds great. Well, thanks again.
Yeah, no problem. Yeah, good. Thanks for having me and great chatting with you. Yeah.
Yeah, hopefully you can come visit.
Yeah, no, eventually. Yeah, that sounds great. Love it.
All right.